In this video, we're going to do some simple cloning by using in silico methods. So at this point, I have Ugene open, and I've loaded some FASTA and Genbank files. Of course, the Genbank files have the annotations associated with them. So this is our cloning vector called puck19, and as we can see, there are a series of features, as well as we have these restriction sites activated, or restriction enzymes activated, and you can see where they are located here. Typically, we're going to use something that's in the multiple cloning site to establish our capacity to insert our pieces of DNA into this space, right, for our control, because the multiple cloning site should represent unique restriction sites within this. Hence, if we look at draw1, we can see there are 3 sites, and notice that they are not in the multiple cloning site, so therefore they're not useful. However, the multiple cloning site, or MCS, we can see here we have some colored in this region here called MCS. Another point of view is here when we look at the linear annotation, there's the MCS, and we can actually scroll upwards, and we can see that they are also within this gene called LAC-Z alpha. And so the restriction sites are listed here and as they're annotated. And we can also look at this annotation down here. If sometimes this is hidden because it's scrolled all the way down, but you can bring it up a little. And what we can see are the unique sites once again: BAM-H1, ECHO-R1, HIND-3, PST1, SAL1, SMA1, XMA1. They all have 1. Draw1 has 3. And we can definitely see, we can rotate this. I'm using a scroll wheel to do this, and we can definitely see that these multiple cloning site has HIND-3 and ECHO-R1 and BAM-H1 and so forth, right? So generally, what we would want to do is identify this multiple cloning site here, and now we can see it's definitely sort of highlighted in this region. We can also go and look at these individual sites here. So now they're annotated, and you can see how they are color coded. So this one is HIND-3. This one over here is just the CDS. This one over here is telling us it's PST1 and SAL1, followed by BAM-H1, followed by SMA1, XMA1, right? Followed by over here ECHO-R1. So in order, we can see ECHO-R1 is the last one, and probably the first one is going to be HIND-3, right? So HIND-3 to ECHO-R1 in this directionality that we see there. So that's the first thing we need to understand about this map, right? The other thing that we need to do is furthermore look at a sequence of interest. So that's when we're going to start looking at something, and today we're just going to look at the spike protein from the Wuhan strain of COVID-19. So this is the original one from 2019, the first one, and this is just a spike protein. Notice that there are no annotations here because this is a FASTA sequence and not a Genbank file, so there is no annotation except for the auto-annotated ones that are based on enzymes. So we have this turned on, and right away we can see that there's an ECHO-R1, there's a HIND-3, there's a BAM-H1. So we have to knock those off. There's 2 PST ones, and that makes it kind of difficult for us to work with, right? Because if we're looking at using any of these here, we're just left with things like SAL1, pretty much just SAL1, and maybe SMA1, right? Is there a SAL1 or SMA1 located? No. Yeah. So SAL1 and SMA1 are not here, but is it in our collection? It's also not in our collection, so that might be another issue. So we may have to actually perform some other types of analysis. So let's do finding the restriction sites, and we may, oh no, SAL1 and SMA1 are there, so they are simply not located there. So that is okay. Okay, so there's some auto annotations. So going further along, we can think about our capacity to perform a digestion and do some sort of sequence manipulation. And what I mean by sequence manipulation is we would not necessarily have just a DNA sequence like this. It would be in the context of another sample, perhaps even an isolate, or in this case, a cDNA. So we can see that it's spike protein and starts with ATG. And if we go to the very end, we can see that it has a stop codon TAA, and doing the math 3822, that is, when you add up those numbers, it's a multiple of 3. So therefore, this is the entire reading frame from start to finish of the spike protein, right? Spike protein gene. So what we can do is actually perform a manipulation and modify these nucleotides simply by adding the appropriate restriction enzyme to them. And the way that we would do that in real life is we would perform a PCR with modified primers, right? So we would basically modify primers. We're not going to do that at this point because that would be a little bit lengthy of a process. So instead, what we're going to do is open up our text editor of this file. And when we do so, we're going to be able to just modify it very simply there. So here's our text editor. On Windows, that would be Notepad. On Mac, it would be something like TextEdit. So we're just going to put in this name just to make it easier for us to identify. And as we see, we just have a series of sequences in our FASTA, right? So this is all it is. And then subsequently, what we're going to want to do is understand what that restriction site is that we want to add to it. So once again, let's go to back to our PUC19, and we'll find where it says SAL1, because once we find SAL1, it is here. And we can see that sequence is GTC GAC, right? So that's what's over there. And so we can GTC GAC. GTC GAC. Right? So notice I put it in a different case just so I can recognize my modification to it. And remember, the FASTA just treats this all as one contiguous sequence, so that's fine that there's spacing issues here. And then the other one that we're going to look at is the SMA1, because that seemed to be working. So that's CCC GGG. Right? So now we would presume that we were able to create some primers that would amplify it and add these on to the end. Typically, in real life, you would actually add a few nucleotides before and ahead. There's actually a chart that you would utilize to determine how these enzymes like to land and cut. So, we're just going to put some ends here. Just for the sake of it, and it doesn't really matter at this point what they may be, but when you design your primers, you would just add a few nucleotides onto the end to ensure that they would fit, right? So we're going to save this as, so I'm just going to call it spike mod because we had modified it, and it'll be easier to find in such a way. So let's go open that, and here's our spike mod. And lo and behold, you can see how we've modified that, right? So this is presuming that we actually had a primer, and the primer would include, some bit like that, as well as our modification. So maybe our primer would look something like that, right? And then at the other end, our primer would look like not this, but the reverse of this, right? So how would we actually figure that out? Well, we turn on the complementary strand there, and we can definitely see what the reverse of that is. So this is starting from the 3' end, going this way, and that would be the reverse primer, right? So since we have that, now we can start to undergo some cloning. So now we're going to start our cloning process by digesting our items into fragments. I was having some sort of error before, so I had to restart Ugene, and once I did, I reloaded everything, and you start to see our restriction sites in our annotations. So remember, we have here a PST1, which we're not using. We have a echo R1 here, which we are not using. We also have here bagel2, which we're not using, and we're not using the echo R5. Nor are we using the Hindi3 sites, which there are multiple of anyway. We are using the ones that we modified at the end, right? So that's exactly what we're going to do. So we're going to go to actions, and we're going to do cloning, and we're going to create our fragments. No, we're going to digest into fragments, I should say. So cloning, digest into fragments, and exactly what we said. We were going to use our sal1, we're add that, and we're going to use our sma1, right? So there we are, we added that, and now we're going to say, okay, so we've made this fragment, right? So it kind of created this new thing here, this new thing being this fragment, right? So that's our first thing. Then we're going to go back to our Genbank file for our puck19, because this is our destination that we're going to put it in. And once again, we're going to clone this into this place as well. So let's just analyze it. Perform cloning and digest into fragments. And once again, we're going to use the sal one as well as the sma one. And okay, right? So right now it's showing it as this sort of circular image here. And then finally, we're going to actually perform the construct the molecule. And here are the pieces that we can use, right? So we're going to use basically, you can see there's multiple fragments, and that's because there's leftover bits from the restriction site in the multiple cloning site, so it's not really that interesting. But here we're going to use the large piece of puck19 as well as the large piece of the spike mod, right? Notice that because this was a circle, we had a digestion here and a digestion here, and we get two pieces. Whereas with the Wuhan spike protein, because they were at the end and they had that little bit of overhang, you had a linear piece of DNA, and you ended up with other bits, right? Because you would have taken a PCR product at this point, and that is a linear molecule. So that's why you get these little bits, because these represent the cut and the overhangs that we had of ends, and then this is the actual piece we want. So we're going to actually add these two there, right? And basically, notice that we are using different ones. We didn't just use sal1. We used different ones because this way it's directional. It makes it a little bit easier for us. And that's important in any scheme, but it really depends on how you're going to do it downstream of this if we're going to modify it in another way. So here we go. We're going to actually, basically perform our cloning. We're going to make sure that we make it circular because it's going to ligate. So in silico, this is what's happening, but in reality, we're going to digest these, run them on a gel, isolate the band in the agarose, extract the DNA from the agarose gel, put them together at a ratio of 3 of the insert to 1 of the vector, and then we're going to put them in a ligation mixture, right, using DNA ligase. So, but here in silico, we're just going to click the make circular because it's going to stick them together. And here we go. Okay. And basically, this is our new molecule, right? It made a new molecule here, and this new molecule looks like this, where we see puck19 fragment 1 here, and the spike Wuhan fragment over there, which is actually kind of large if you look at it, right? But that's what it is. It's a very large thing. So at this point, now we know that this is what we would actually be putting into our subsequent usage for carrying it in bacteria. We'll transform this into bacteria and hopefully be able to propagate it. One thing that we want to do is actually rename the sequence, right? Always go to this little DNA molecule and rename the sequence because this is our puck19 spike. And so there it's labeled more appropriately. We do have this over here with the annotation in this file, and we can perform some more edits on the Genbank file just to make sure that this is labeled appropriately. But afterwards, what you're going to do is, ensure that we are going to utilize this in a constructive way. So here's our Wuhan. We edit that, and so now it no longer says it's that fragment piece. And even here, we can remove this labeling because it's not important for us to know that anymore. So let's disable that. And then this looks more like what we're expecting. We have an origin, a replication, the selection marker of ampicillin resistance, and then here in our multiple cloning site, we still have remnants of the multiple cloning site there, of the sites that we did not use, and we can see that our spike protein is in its place. Notice something that is missing. We no longer have that LAC-Z alpha gene listed here. It's still there, but the thing is we've disrupted that gene, and therefore it's not in existence anymore realistically. We can still find it, but it's not going to be functional, right? So here we have our inserted and cloned spike protein from the Wuhan virus. Okay, so we're going to now clone this into our shuttle. There's an issue with our shuttle. If we look at it, I know that there is a SAL1 site here. We also know that it's supposed to look for SMA1, XMA1 sites, and for some reason it's not being annotated. So we're going to have a problem with that in silico, right? But here it is. We can see it right here, CCC, GGG, which is our SMA1 site. So for some reason it's not detecting it with the program. I don't know why that is. Some weird bug, but we'll find a way around that to modify it. And so that's what we'll do now. We're basically going to take this and we're just going to digest it into a fragment by cloning, digest into fragment. And since we don't have the option of using SMA1, we're just going to use the SAL1. We're going to add that, and here it is, right? So we have this new a new molecule once again, and actually what I want to do is further export this, right? So I just right-clicked on it, or command-click, I suppose, and essentially what we're going to do is try to take this fragment and actually export it to this sequence to a new FASTA sequence, right? So we're just going to say this is the shuttle fragment, and we're going to say SAL1, just so we know, and we're going to add this here as well. So here it is, it's showing up. And once again, we can see a bunch of our restriction sites over here. We can turn on the annotation. We can see BAMH1, Bagel1. It does not see the SAL1 site anymore because there's that overhang. It doesn't exist anymore because it's a linear thing. We're going to turn that off just to make it a little bit easier. And then basically, we're going to do a search for our CGG, and we're just going to make sure that we find that, right? So our SMA1 is CCCGGG, and it's right over here, right? So that is where we see it right there. So if that's the case, we do know that it is going to be cutting from here, right? At this GGG, and we will select this all the way back to the end as we see it, right? So this is basically the other end, just so we can understand the frame of reference. And actually what we want to do is remove that first bit, right? So I just wanted to show you that location. We see the CCCGGG. So we're actually just going to go and remove this. So let's go to edit mode, switch on editing, and we're just going to go to try to remove these, but it's not letting us. So let's see. Yeah, since it's not letting us, we'll go the other way, which is this way. And once we have done that, we've selected the bulk of it. We've just removed that bit that would have been cut out that we're not interested in between the SAL1 and the SMA1 site. So we're going to export this region again. And so now we're just going to say this is SAL1 SMA1 site. Export that. And let's hope this works, right? So similarly, we want to go back to our other document, which is this plasmid here, and we're going to subsequently perform a cloning by digesting this into the appropriate fragments. Here we are lucky enough that we see these, so we'll add those two. Okay, and so now we have this fragment, and we can see that once again there are two fragments. The fragment that is actually the Wuhan, and the fragment 2, which is actually the PUC19 fragment. So let's go to cloning and construct our molecule. And now we have a bunch of options here. So we're going to go with the pShuttle fragment, and it's not letting us have it. Okay, so that's the pShuttle fragment, which is this fragment, and it's not showing up. So that's unfortunate. Okay. Okay, so since Ugene was once again having issues with this, I simply created the selection of the Spike SAL1 SMA1 fragment, which is this, and exported it as a FASTA sequence. We can see at the beginning there's the last C of the SAL1 overhang, and then when we go down to the bottom, we should see the CCC of the SMA1 over here, right? So that's really what the fragment should look like. And then the pShuttle fragment we already made, and I basically exported it as a separate thing. Once again, here now we can see the end is GGG because that's the SMA1 end. And then we go down to the bottom, and we can see that there's this overhang here of the CTTG, and then there would be the GCTTG, and then C would be the overhang that was cut off from the SAL1 end. So we know that we have these two FASTA sequences. So since Ugene doesn't qualify and help us in this situation, we have opened these two up in our text editor. And I know that this is the insert, so if we go to the very beginning of it, we can simply see that it is what it is. And I'm just going to double click on this line, and I'm just going to copy that, and then I'm going to go to the very end of this, and we are going to add that, right? So now it's been added, and I'm going to save it as something else. As that, well, that's not good. pShuttle. Okay, so that's really what I should have done, and I should rename it here, SMA1, SAL1 to Wuhan Spike, right? So let's just save that again, and let's load it here. And the problem now is it's lost the annotations when we've done it because we used the FASTAs, but that's the easiest way to do it. So what we're going to do is simply open up this file and look at it. And yes, here it is. It's just a sequence with no annotations, but we actually do know something about it. We can mark it as circular, and now we have the beginnings of our diagram that we can add back our annotation here, which requires us to do some work, but it's a larger plasmid. It's 10434, and we can go and also look at our original pShuttle and start to think about whether or not we can search for this, and we can start to even look at these regions here. So let's just look at this, and we can select this sequence region, which is for the origin. Okay, and so that's our selection there. We can go back to our unannotated item here. So here we are going to go search for this bit, and as you can see, it actually has found this origin located in this space, right? So that's important, and we can actually go here and think about adding our annotations, and to start over with adding these sort of useful portions of this that we really want to know about, right? The other thing that we can do otherwise is perform another analysis and actually just look for common things that we really want to know about in these sequences. You can look for the ORFs, you can find the annotated regions, right? You can do the annotation of this plasmid once again to actually perform and create the appropriate annotations, right? So here are the automatic ones. So let's go automate them. And so it caught some of it. It did not catch this here, which we want to annotate. So here's our annotation. And for this, we know it's an origin. So we have to find the replication origin right there. We can rename it whatever we want. Create that, and once again go back here and export another item that's probably important. So this one is important, so we're just going to double click on that, and you see how it's selected. We're going to select this here. Oh, select the sequence region, sorry. Copy that, go back here and search for that, and it's over here selected, and so that was our kanamycin. So we might want to actually go and once again edit and annotate that. And for this, it was a selection marker or antibiotic resistance. Or we can even call it a gene, right? So we have to look through here. I don't think those are available, so we're just going to call it a CDS. So we'll just call it a CDS. And this CDS is going to be called kanamycin resistance, right? So edit. And one other thing, oh, that did not do the right thing. So that was supposed to be the replication origin. So now that we've gone through the trouble of annotating this FASTA sequence, we have a FASTA sequence still by itself, as well as a Genbank annotation file, right? I've actually removed the other auto annotations, which just by disabling them, there's no need for them anymore. And these are the ones that are going to be saved. I've saved them to a place where I can find. I actually made a copy called pShuttle Spike GB, which is actually what the final is going to be called anyway. And so the way that you do this is you go to merge these two things together, is you go to Workflow Designer, and you can create a workflow very easily. So what we would actually do is take a sequence, drag it out here. We would also want to read in an annotation and drag it out here. And then we'd want to multiplex those things together by doing this. And then we want to write it to a sequence, right? We'd have to adjust all these things that are unset and so forth. But there's an easier way to do that because it's a common occurrence. You go to Workflow Designer, you go to Samples, and under Scenarios, there is a Merge Sequences and Annotations, and it goes through this process here. So we're going to double click on this to load our samples. It wants our sequences, and our sequence is going to be called pShuttle Spike FA, and our input annotation is going to be called pShuttle Spike here. And it's asking us for results file, so and that's there. It's going to actually put it in a really obscure place, unfortunately. Just make sure you know where it is. It's going to put it into a home directory on a Mac. That would be your users, whatever your name is, and there would be a separate folder called workflow run output, one called report, and one called pShuttle Spike GB, which is where it is. So that's where I just gotta know. It's unfortunate that it's very difficult to reset it to a new place for unknown reasons, but here we go. We're gonna actually run this. Don't apply it, run it, right? So when you run it, it goes through this, and this is the output, and basically it has read something, it has read another thing, it's multiplexed it, and there's no output message. It just happens to be something that it writes for us. So now we're going to actually go into my file system, and I am going to look for this in my home directory, which is off screen at the moment. So looking in my home directory, into my workflow output folder, it is the one that has this time, and here I go dragging it in here. And one thing that it did not do properly is it is not a circle. Now, one thing that we can do subsequently is actually edit that fairly simply. And so here we're going to go to this, and basically we're going to open up that file. Just let me find it. And ooh, that's dangerous. Right now I opened up this one, and all I see is this. So that's not the right one, which is good, right? It's actually in my home directory. So let me open up the correct one. And once I open up the correct one, it'll make more sense. It's my workflow outputs. And here it is, right? So this makes more sense. This is the correct file. And so what we do is I had the original pShuttle open, and I'm just going to copy that because it says that it's double-stranded DNA and it's circular. And that's what we want to know, as well as the time that it's generated. And there we go. So let us save that, and right away Ugene wants to redo this. Yes. And now it is permanently circular, and that's exactly what we want to see. So when you can have this, you can pass this on to someone else and say this is the pShuttle vector with our Wuhan spike protein in it, and it's set to go where you co-transfect this plasmid with the AdEZ plasmid into 293 cells, and you'll start to see those 293s dying as adenovirus is produced, and we will have functional adenovirus that we can purify for the sake of using in a vaccine if we wanted to.